We're most familiar with photosynthesis because it's what our plants do. Now, plants are more complicated organisms, but what plants are doing is capturing the sun's energy and using it to build their own food, glucose, sugar molecules. They do so from simple ingredients in their environment, carbon dioxide in the air and water molecules. As a result of this chemistry, oxygen is liberated into the atmosphere. So while we're most familiar with photosynthesis in plants, plants didn't originate photosynthesis. But let's take a look at plants briefly in more detail. Plants are multicellular, so if we take a leaf here and we put it under the microscope, we'll see these units, these brick-like units. These are the plant cells. And of course, they have these green structures in them. The green structures in them are called chloroplasts, and this is the site of photosynthesis. The cell wall here, these are rigid boundaries of the plant cell. The cell membrane is just inside the cell wall. Here we see the cell nucleus. And of course, the cell is filled with fluid, the cytoplasm. But it was bacteria that invented photosynthesis long before plants evolved. So we're going to credit bacteria with photosynthesis. And in the next part, we'll take a look at exactly what is photosynthesis. Now, what is photosynthesis? This is the overall reaction. We've got the reactants, or the starting materials, on the left, and then on the right of the arrow we have the products. The whole point of photosynthesis is to produce this molecule right here. It's glucose. It's a sugar. We can think of it as food. So what the cyanobacteria is able to do is to convert light energy into the chemical energy of food. They can make their own food. Again, the two simple ingredients are carbon dioxide and water, and these are very common molecules in the environment. So no longer then did the cyanobacteria have to eat other types of nutrients. They just needed a steady supply of these two, and they could make their own food. Here we see the cyanobacteria in the middle. They're making glucose, and here we see the molecular structures of the water molecules and the carbon dioxide molecules. Here's glucose here. Light energy is powering this chemistry, and a waste product of the chemistry is oxygen. Now, the photosynthesis equation then obeys the conservation of matter. So during chemical reactions, atoms are not created or destroyed, only rearranged into new molecules. The chemical bonds of the reacting molecules are broken, and new bonds are made to create the products of the reaction. In photosynthesis, energy from sunlight is used to break the bonds of the starting molecules and create new bonds in the products. So if we counted up all of the carbon atoms on the left side of the arrow, we would see there are the same number of carbon atoms on this side as there are on the right side. That's the conservation of matter. Let's take a look at carbon. There's no carbon atoms in water. There are carbon atoms in carbon dioxide molecules. There's one atom per molecule, but we have 6 carbon dioxide molecules in this reaction. So we have a total of 6 carbon atoms on the left side of the arrow. Over here on the right, there are no carbon atoms in oxygen. There are 6 carbon atoms in one molecule of glucose. So again, 6 equals 6. And we could do the same for the other atoms, and we see that the number of oxygen atoms on the left is equal to the number of oxygen atoms on the right. The number of hydrogen atoms on the left is equal to the number of hydrogen atoms on the right. That's the conservation of matter. Atoms are not created or destroyed. They're simply rearranged during these chemical reactions. In addition, we can think of photosynthesis as a conversion of energy forms. So during photosynthesis, solar energy is converted into chemical energy. And this is important because solar energy cannot be stored or transported. Glucose molecules, with the energy they contain in their chemical bonds, can be stored for later use within the cell. And they can also be transported between cells in later multicelled plants. So one of the important features of this reaction is that the cyanobacteria evolved the ability to convert one form of energy, solar energy, that cannot be stored or transported, into another form of energy, chemical energy, the energy stored in chemical bonds. And this molecule can be stored and it can be transported. And that's very valuable. Now, we said that this chemistry produces waste. The oxygen is considered a waste product of the chemistry. The whole point of photosynthesis is to make the sugar molecule, and this is leftover stuff, as it were. So we'll call this a waste product of photosynthesis. Of course, it's not a waste to us, animals. We need this oxygen. But for this chemistry, the oxygen is a waste product. And specifically, the oxygen that is released during photosynthesis comes from the water molecule. Notice both carbon dioxide and water molecules have oxygen atoms. It's the oxygen atoms in the water molecules are the ones that are released. So what the sunlight energy is doing, it's breaking apart the water molecules. The hydrogens and all of the atoms in the carbon dioxide molecules will be used to make sugar, and the oxygen atoms in the water will be released as the waste. If we put a plant, an aquatic plant, and illuminate it with some light and put a sort of a test tube over it, we can collect these bubbles as the plant grows. These bubbles are the liberated oxygen. So we can actually see the result of photosynthesis in the formation of oxygen bubbles when you shine the light on the aquatic plant. Of course, if you turn the light off, then the plant has lost its source of energy and it cannot perform this photosynthesis reaction. So you turn the light off, you will not get the production of these bubbles of oxygen. You turn the light on, you will get bubbles of oxygen being liberated as waste from the photosynthesis reaction. So what is the plant or the cyanobacteria doing with this light? Well, it's absorbing some of the light to power this chemistry. So we can think of this then as a process where the cyanobacteria or the plant has to absorb some of the light. And that's the source of energy to be used to build these food molecules. Well, the fact that cyanobacteria are green suggests that they reflect green light and absorb the blues and the reds. So let's review what we know about light. So stars produce white light. We can separate the white light into the colors of the rainbow here using different instruments, a prism or this spectroscope we'll see in a minute. And the fact that cyanobacteria are green suggests that the green light in the white light is being reflected, suggesting also then that the blues and the reds are being absorbed. Now, the reflected green light cannot be used as an energy source, so it suggests that the cyanobacteria are using the absorbed colors, which would be the blues and the reds. Now, let's imagine our setup here again. We have some cyanobacteria here, or plants, and we have a light source here, but let's imagine we can change the color of light that we shine on the organism. Well, changing the color of light then will show which colors produce the maximum rates of photosynthesis. And we would predict that green light, since it's reflected, cannot be used. It's not being absorbed, so the green light is going to show a low rate of growth. In other words, the cyanobacteria and plants will not grow if you just shine green light on them. They reflect the green light. They can't get the energy of the green light because they're reflecting it. And on the other hand, we would predict then that the blues and the reds, the light that is being absorbed, would produce higher rates of growth. So let's see. Here is our graph. We have the y-axis, the relative rate of photosynthesis. And by the way, we can measure photosynthesis by measuring the production of oxygen. So for every food molecule that is produced, 6 oxygen molecules are liberated as waste. So if we can just measure the amount of oxygen produced, that's a measure of the rate of this entire reaction. And that's what our y-axis is here, the relative rate of photosynthesis. X-axis is the different colors of light that we might shine on the plant or the cyanobacteria. And sure enough, we get the highest rates of growth for the purples and the blues. We get low rates of growth for the greens and the yellows. And we get high rates of growth again if we shine red light on the organisms. Now, why is this? Well, if we look at the surface of the cyanobacteria, we're going to see these important molecules, these pigment molecules that are anchored in the membrane of the bacteria. And they are called chlorophyll molecules. They're pigments that can absorb the blue and the red light, and they reflect green light. In fact, for green plants, you can do a simple experiment. You can put a green leaf in some alcohol and boil it, and that extracts the chlorophyll out from the leaf cells, and you can get a tube filled with alcohol and chlorophyll molecules, and it looks green. The solution has turned green. Well, now then we can use our spectroscope to examine the absorbing properties of chlorophyll. So again, a star produces white light, or a light bulb in a classroom, white light. We'll have all the colors of the rainbow. The spectroscope is this device right here that will separate the light by wavelengths. Now let's do a simple experiment. We'll compare, looking at a white light source. We'll see all the colors in our spectroscope, but now let's see what happens when we put chlorophyll in front of the device. What we see is the blues and the reds disappear. Up top here we see white light, and we see all of the colors of the rainbow. Down bottom here we have put the green test tube filled with the chlorophyll, the green chlorophyll, in front of the spectroscope, and now the blues and the reds have disappeared. We only see the green. So the chlorophyll molecules are reflecting green light, in effect allowing some of it to go through the test tube filled with chlorophyll. Here's another way of looking at this. On the right we see again white light separated into all the colors. Again, we put the tube filled with chlorophyll in front of the spectroscope. The blues and lots of the reds are disappearing. And if we look now at an absorption spectrum of the chlorophyll molecules, there are two types of chlorophylls, A and B. That won't concern us here, but this graph then is showing the peaks will show the amount of absorption of chlorophyll for the different colors. So we have high absorption for the violets and the blues. So the chlorophyll molecules absorb a high percentage of the blue lights and a significant percentage of the red lights as well, red colors. And so that's why we get the disappearance of the reds here and the disappearance of the blues here. The chlorophyll molecules are absorbing selectively the blues and the reds. The greens, you'll recall, are being reflected. So if we think of a bacterium, then we want to think of how it interacts with light. We've got white light hitting the bacterium. The chlorophyll molecules on the surface are absorbing the blues and the reds, and that's going to be the energy to power the chemistry inside the bacteria cell. The green lights are reflected. So this is selective absorption. And the absorption by chlorophyll is what explains the growth rate here. So the peak of absorption is in the blues and the reds. These are the colors that get maximally absorbed. They are also exactly the colors that produce the highest rates of growth. And that's not an accident. The absorption of this energy is what powers the photosynthesis reaction. So we get these two graphs have peaks in the same colors, because this graph up here is explaining this graph down here. The absorption of the reds and the blues is causing the greater growth rates in the red and blue light. In summary then, cyanobacteria evolved the ability to perform photosynthesis using two simple ingredients in their environment, carbon dioxide and water molecules, to make sugar molecules with oxygen liberated as a waste. This allowed cyanobacteria to make their own food, and so no longer did they require other kinds of nutrients in their environment, nutrients that would have been getting more and more scarce as bacteria populations increased. So natural selection then favored the evolution of new adaptations, new molecules to make a new way of life, to capture sunlight energy and make your own food.